Hello, my name is Julieta Starker, and I will be talking today about the optic flow processing centers that we have discovered in the crabs and how they are very similar to the ones already described in flies. All visual animals have to deal with two different aspects of the image at the same time. One has to do with the detection of particular objects that can be prey, predators, conspecific, etc. And the other has to do with the movement of the whole panorama that occurs when an animal moves. And this is called optic flow. For example, if I move forward, the whole panorama will move backwards. In the lab, to study this type of processes, we use the coherent rotation of the panorama. In that way, we can control the speed of the stimulus, the contrast, etc. The presence of a panoramic movement in the retina causes the animal to perform compensatory responses that can be driven by the eyes, by the neck, the whole head, or the body, and that try to focus the images in the retina of the animal to reduce the blur. These are collectively called optomotor responses. These responses also assist in navigation. For example, think of a fly trying to fly in nature from point A to B, and in the middle, a gust of wind makes the animal turn. And so the visual system of the animal will experience an unexpected movement of optic flow that will lead the optic flow processing center that has directional neurons involved in horizontal or vertical stimulus to connect with motor neurons in the neck or in the flies or in the wings of the fly to perform corrective optomotor responses that lead back the animal to the original route. In the case of crabs, we can see strong optomotor responses led by the movement of legs or the eyes. In this case, we present the movement of an actual drum, high contrast drum around the animal, and you can see the animal trying to rotate at the same speed of the stimulus to compensate the movement in the eyes. When you analyze the movement of the eyes, on the other hand, they cannot rotate 360 degrees. So they had to make a slow movement trying to compensate the movement of the stimulus, and then a rapid reset. This is very similar with our own optokinetic nystagmus. And in this example, you have the animal surrounded by a panoramic stimulus that is moving this way. And you will see that the eye tries to follow the stimulus slowly and then resets back and then moves slowly again to follow the stimulus. This type of graph will then detail how the eye is moving and represent a slow phase and a rapid phase, as in our case. Until very recently, we didn't have any clue of where in the nervous system of this animal was optic flow analyzed. Because we know that the optic lobes of the crabs contain the optic neuropiles that are very commonly organized with the ones in flies. And this is all the nervous system that processes the visual images. And they are organized beneath the retina as containing a lamina, medulla, and lobula connected with two chiasmata. This is a rather difficult to explain the presence of two chiasmata without a common origin, and that's why the neuroanatomy of the optic neuropil has been used to construct phylogenetic trees inside to explain the relationship between different groups in arthropoda. In the fly, the third optic neuropile, the lobular complex, contains not only a lobula, but also a lobular plate that has been extensively studied, in particular because of its role in optic flow analysis and in commanding optomotor responses. this structure, it has big tangential neurons. They have about 60, containing two systems of directional cells responding to horizontal or vertical displacement of the images. And the lobular plate has 4 layers. Each of these layers respond to one of the 4 cardinal directions. And it receives inputs from the medulla and from the lobula. Because of many different alterations that have been done in the lobular plate and the optomotor responses were lost, we know that this structure is central. So in the crabs, we have recently identified a small neuropile that is placed in the right position as to be a candidate to be a lobular plate in crabs. They have similar connections with the medulla and with the lobula. And we noticed that this lobular plate is smaller in crabs than in flies compared with the size of a lobula. But of course, if you think of a fly that flies very rapidly and it can move in 3 possible axes, yaw, roll, and pitch, the optic flow that the visual system experiences is rather complicated, especially if you compare it with the natural environment of our species of crab, that is a semi-terrestrial crab living in a very flat environments, where when it moves, it actually experiences mostly horizontal optic flow. Then the processing system for optic flow have to be simpler than in flies. We started by analyzing the connections of medulla and lobula with the lobular plate. And for doing this, we use the injection of small dextrans with different colors to study the retinotopy of these projections. And we found out that both the medulla and the lobula project in an orderly way, a retinotopic way, into the lobular plate. And most interestingly, the arborization of these inputs organized in 4 different layers. So because we know from the histological preparations, for example, this is a Bodian staining of the lobular plate, and you can see here a huge tangential neuron in this structure. We know that these neuropile in crabs have 3 properties in common with flies. They have arborization cells, they have retinotopic inputs from the medulla and from the lobula, and they are organized in 4 layers. So all this led us to a question of if we have so many morphological attributes in common with the fly, would this structure be doing the same role than flies? So would it be involved in the performance of optomotor responses? So to deal with this question, we decided to do electrolytic lesions and ablate this particular area and compare the behavior of the animals when compared with animals that were lesioned in other parts of the optic neuropiles or with control animals without lesions. The results show very strikingly that animals that were lesioned in the lobular plate display a much poorer optomotor response. On one side, many of the animals did not respond at all to optic flow. And you can see here that the responses, about 40% of the animals only respond to the panoramic movement, while control animals respond about 100%. But interestingly, even those small amount of animals that responded to optic flow, they showed asynchronic and poor responses. Here you can see the number of tracking cycles in 3 minutes of stimulation, and you can see the lesion of the lobular plate produces that about 2/3 of the cycles are lost. And the cycles that are remaining are slower, so they are not actually tracking the image, but they are staying behind. So there is a poor tracking of the image and the compensatory response is not done well. Importantly, this impairment was specific for optomotor responses because when we measure the response of the same animals to an approaching stimulus that could be dangerous for the animal, they elicit the same type of anti-predatory response in control groups and in lobula plate lesion animals. Both the amount of animal responding and the velocity they display in their escape responses were equal between the groups. So this is very important because it's the first functional evidence that the lobula plate of crustaceans and dipters have the same role. More interestingly also is that we found some years ago we made some experiment occluding the vision of one of the two eyes and measuring the response of the animals to the movement of the panorama in the two possible directions, clockwise or anti-clockwise. And while binocular seeing crabs responded equally to both directions, when we occluded the vision of one of the two eyes, something really striking appeared. That is that animals will only respond in one direction, and the response was completely lost, and even anti-compensatory responses were obtained in the opposite direction. If the other eye was occluded, then the response would be in the clockwise direction. The two of these conditions have in common that they are stimulated from front to back, the eye that is seeing, the seeing eye of the animal. So this is really interesting when compared with similar experiments made in flies, where you can see here that front to back motion elicits a really big optomotor response in flies, while back to front stimulation makes animals to perform anti-compensatory responses or even null responses. So in flies, we know that this type of unilateral optomotor response has to do with the fact that there are specific neurons in the lobula plate that are responsive for the horizontal system, and they respond much more strongly to front-to-back motion than to back-to-front. So the fact that we are finding exactly the same kind of responses in crabs makes us propose that probably the underlying circuit is really similar in the two groups of animals. And if we record in the lobula plate, it will be very probable that we will find more neurons responding to front-to-back motion than from back to front. So in conclusion, we have proved common morphological traits and function of the lobula plate in crabs and flies. The size of and complexity of the neuropile varies according to the ecological needs. However, many of the similarities, like the behavior of the monocular animals that I just showed you, are not easily explained, but considering a common circuit that might possibly be inherent. So with this, if you have any question, I'll be happy to answer it. And thank you all for listening. And thank you all the researchers that were involved in these experiments, especially Shair that did the lesions of the lobula plate. Thanks also the grant agencies for supporting work. And again, for you for listening.
Okay, thank you, Julieta. Amazing talk. Now it's time for one question. Let me see on the chat. Okay, when you perform the electrolytic lesions in the lobular plate, you lost the optomotor response around the 40% in contrast to the high 100% of control group. However, I also noticed that the control lesions group display a little less optomotor response than the control group. Did the amount of response depend on the exact location where the lesion was applied?
Oh, thank you for the question. Yes, if I can share my screen, I can show you. When we did these experiments, of course, they were not a specific place where we were aiming, but afterwards, when we analyzed the data, we did notice that the amount of response depended on where this control lesion appears. If you can see here, if the lesions were in the more peripheric neural fields like the lamina or the medulla, the response was always high, but there were some spots that here are seen in red that produce a marked reduce in the optomotor response. We think that this has two possible explanations. One is that we are making a lesion in one part of the connected circuit with the lobular plate. For example, in the lobula, it might be that the inputs coming to the lobular plates are being damaged, or when we see this point here, that might be the track of the lobular plate going down. On the other side, these lesions that are producing a reduced response might be related with other center that we have found. That we found some neurons in the lobula of the crab that are also directional, and that they all project to a center that is about in the right position to be modified by a lesion here. So I think that it might be possible that in some of these cases we are also affecting other parts of the network that is processing directional motion.
Okay. Now, I have one more question, or you prefer at the end, or I don't know. It's short.
I can.
Okay. Francesco Mattia said that Pusky showed that the optomotor response in rat and mice monocularly deprived of vision appears only in one direction, just as you showed in flies and crabs. Do you have any idea of why?
Well, the interesting thing is when this unidirectional phenomena appears in several animals along all the zoology scale, but when you see it in vertebrates, it appears in the opposite direction. So vertebrates will respond to movements from back to front, and arthropods from front to back. And my guess, and it's what is seen in flies, is that it has to do with how the animals more often locomote, and especially on how are the properties of the directional neurons that are in the brain processing this. So it was really striking when we got these results, and I found out that in vertebrates, the direction was opposite. So I guess it has to do on how the directional network functions in each of these animals.
Okay, well, thank you, Julieta.
Now,
it's a pleasure for me to present the second speaker, Flavio Solesi. Flavio is a doctor of biological science, PDCIVA, Uruguay. His current positions are Associate Professor, Cell Biology, School of Science, University, Uruguayan Republic, Investigator Grade 4, PDCIVA, Uruguay, Investigator Level 2, National System of Researchers, Uruguay, and Associate Researcher, Institute Pasteur de Montevideo. He has published 17 papers in the last 10 years as corresponding author. His research is focused on the relationship between cell polarity and neural differentiation in vertebrates, concentrating on polarity transition processes such as those found during the neurulation and neuronal differentiation. The title of his talk is Crossroad Blues, the role of SLIT2 in the development of the zebrafish optic pathway. Flavio?
Hello, my name is Flavio Salesi from Facultad de Ciencias, Universidad de la República. I'm going to tell you today about some recent results from our lab done by Camila Davison during her doctoral thesis, soon to be defended in October. The talk had to do with decisions. Decisions like the ones Robert Johnson was singing to in 1937. He was referring, of course, to decisions in life, in human life, but there are other important decisions, like the decisions that neurons have to take to eventually grow through commissures in the brain. And one of the most well-known commissures since antiquity is the optic chiasm, here in a drawing by Newton from the early 18th century. And he realized that in the case of the human, because of stereoscopic vision, we needed to have roughly half of the axons crossing at the level of the optic chiasm and half of the axons growing ipsilateral. In that way, the two fields will be separated in the two hemispheres. But this is not the rule for all the species. We have different situations depending on the species we are looking at. And for example, in the zebrafish, which is our subject of study, we have a complete crossing of the optic axons at the level of the chiasm going towards the contralateral optic tectum, which is the target. How is this achieved? Essentially, we know now quite a good deal of information about the growth of axons, of the optic axons, but still there are many mechanisms we don't know. But we know that the path is rather long, and the axon have to cross many situations to eventually arrive to the tectum. And one important one is the optic chiasm. And here, a signal that is important is the SLIT molecules. SLIT molecules are secreted proteins, rather large proteins, which interact with heparan sulfate and also with receptors at the cell membrane, which are the roboreceptors. And these receptors transduce a signal towards the cytoskeleton, affecting particularly actin dynamics. And this usually has the effect of repulsion over the growth cones that are affected by the signal. Hence, we were interested in looking at this signaling in SLIT signaling in the retinal ganglions growth, in the axon growth in the zebrafish. Camila looked first at the expression of these more important genes, and particularly we were looking at SLIT2 and ROBO2, thinking that SLIT2 would be a good candidate for being a signal for axon guidance at the level of the optic chiasm. And indeed, SLIT2 is expressed all along the optic pathway, and these are the optic nerves, and also here the optic chiasm, and all along here we have expression. And also ROBO2 is the main robo expressed at the level of the retinal ganglion cells at the right time. So this could be an interesting pair to look at. Camila started by doing a mutant, generating a stable mutant by CRISPR-Cas9, using guide RNAs directed towards the initial part of the protein, of the gene encoding the protein SLIT2 in the zebrafish. And eventually, from all the mutants that were generated, one was very interesting because it had a 10 base pair deletion very close to the initial ATG. And we see here how this mutation will lead to frame shift in the reading sequence, and hence a very short version of the protein, which is completely nonsense, essentially in amino acid sequence. Hence, it's a known mutant. In these mutants, however, there is no external phenotype that can be seen in the embryos, not even in the maternal zygotic mutants. However, we wanted to see what happened at the optic chiasm. So one thing we wanted to know initially was how are these optic chiasms formed? What is the normal shape of the optic chiasm in this species? This can be achieved by, for example, labeling with tracers from different eyes, lipophilic tracers like the eye and the O, which Camila did here, injecting in one eye the eye, in the other eye the O. And we see here how these optic nerves are completely labeled by this staining, and we see how at the level of the optic chiasm the nerves cross without actually mixing, and without mixing of the axons at this level. So they are completely segregated. Here, one crossing more anteriorly than the other, but very close together. What happens in the mutant? And this was a very interesting surprise, where we found very usually that one of the optic nerves was split in two, and the other optic nerve was going through the hole left by this split nerve at the level of the optic chiasm. And this phenotype was rather penetrant, having more or less 40% of embryos with phenotype in the homozygous, and also after treating the embryos with morpholino, so the morphand was also had a very nice phenotype, very similar to the one seen in the mutant, and I will show you results from both of them, mutants or morpholino treated embryos, depending on the experiment. Here, for example, it's an experiment done with morpholino, where Camila wanted to see what happened at the very first moment when axons were reaching the optic chiasm, and we see here how they behave in the normal embryo, where they just cross very cleanly, and they start to form the optic chiasm and the optic tracts, and how they make mistakes at the in how the mutant axons make mistakes at the level, eventually leading to this split optic nerve we see here. We will see the movie here again, and here is how these axons are making these errors that eventually could lead to the phenotype we see. So, is this the whole story? Is there anything else? Is there anything else happening? What is going on here in each optic nerve? And one thing of looking at this is to now inject the eye and the O in different regions of the retina, and try to see if there is a differential segregation of axons. In the normal embryos, we see that axons from temporal or nasal retina are completely segregated all along the pathway, and here will be the optic chiasm in this dotted line. The optic nerve has a segregation, the optic chiasm has a segregation, and also the optic tract all along the pathway. And this is seen here in cross sections and also in these quantifications. What happens in the morphans, in this case in morpholino treated embryos, is the same in the mutants. We see that there is a segregation at the level of the optic nerve, which can be seen here at the cross section, but at the level of the optic chiasm, already at the level of the distal optic nerve, then the optic chiasm, and also the proximal optic tract, we see a complete mix of axons coming from both regions of the retina, to eventually be corrected at the level of the distal optic tract, where again we see a segregation. So we see an apparent correction of these errors made at the level of the optic chiasm in the distal optic tract. What happens in the optic tectum? At the optic tectum, we would expect to see a complete segregation of innervation, retinotopical distribution of innervation of the axon from the retina, where they are distributed in this way with quadrants that can be very easily recognized. So what happens in these mutants? Essentially nothing. So we see that there is no phenotype at all. The morphans and the mutants all have a very correct segregation of innervation at the level of the optic tectum after labeling with the eye and the O. Hence, the phenotype seems to be concentrated at the level of the optic chiasm. Now the question is this the complete story? Is this the whole story? And some things tell us that it is not. One thing is that the phenotype of the ROBO2 mutant, which has been known by more than 20 years now, is much more severe at the level of the optic chiasm. And this was seen by Qian's lab at 5 days post-fertilization, and we also see it here in this Camila's experiment much earlier, as 48 hours, where we see some axons which are completely lost, not simply a split of the optic nerve, and they can grow ipsilaterally, and we don't see that with the SLIT2 mutants. So why is the reason for that? What's the reason for that? One reason could be that eventually there are other slits involved. And indeed, this is what has been seen in the mouse, where SLIT1 mutation, SLIT1 and SLIT2 are two slits expressed in the mouse around the optic chiasm. We see that SLIT1 mutation doesn't have any phenotype, as happens with SLIT2, nothing that can be seen at least morphologically very easily. And the double mutant, on the contrary, has a very strong phenotype, quite similar to the robo mutant in the zebrafish, with what they call a double optic chiasm. And this can be seen here also in labeling with II and IO. In the zebrafish, we don't have SLIT1 expressed around the optic chiasm level, but yes, we have, but we do have SLIT3, the other slit. So we wonder if SLIT3 could be collaborating here with SLIT2 in the correct crossing at the optic chiasm. So Camila, what she did was to now use CRISPR-Cas9 technology to inject embryos that were in the SLIT2 background, so SLIT2 maternal zygotic mutants, which we know in a great proportion have this split optic nerve. So this is a phenotype we see here. He injected SLIT3 guide RNA together with Cas9. When doing it alone in a wild type background, there is no phenotype. So SLIT3 doesn't seem to give any phenotype at the level of the chiasm. However, the double mutation, SLIT2 and SLIT3, gives a phenotype which is much stronger and very similar to what is seen in the Australian, in the robo2 mutant, indicating that probably both signals, SLIT2 and SLIT3, are collaborating, acting probably through robo2 to allow these axons to properly cross at the optic nerve level. This is even more clearly seen here in the II and IO labeling, where we see ipsilateral terms, where we see axons going towards the other eye, and even lost axons completely from both retina. So the phenotype is much stronger here. So as a summary, what I've shown you is that the SLIT2 gene is expressed around the area in the zebrafish, around the area of the optic chiasm. Also, it is in the retina. I didn't show you this, but we didn't talk about the retina today. And here we have a very nice, interesting phenotype, rather complex phenotype, mostly involving segregation of axons at the level around the optic chiasm in the SLIT2 mutant. In the cases of SLIT3 mutant, we didn't see any phenotype, so it doesn't seem to be so important alone for axon crossing at the level of the optic chiasm, but it seems to be collaborating with SLIT2 because the double mutant has a much stronger phenotype, very similar to the receptor phenotype, indicating then that they both collaborate. Finally, just wanted to tell you that we are very happy that a great part of the results have been published in Cells and Development. It's the second issue, and even it is the cover of the issue. So that's a very, very interesting news for us. And I want to thank you all, thank our collaborators, and all the group members, and particularly Camila, who was the one making the experiments I just showed you. And with this, I finish, and I thank you for your attention.
Well, thank you, Flavio. It was an outstanding presentation. I have one question. There is a SLIT2 expression in the retina. What cells are expressing it, and does it have a role in retinal differentiation?
Thank you, Euge. I will just share my screen here. Yes, if you can see the presentation here, let's hide this and maybe move this a little bit. Yes, we see expression from around 40 hours post-fertilization at the retina, and this expression. So actually, there is an expression also earlier that comes from the CMC, so the ciliary marginal zone. That one starts to disappear later. And what remains is an expression in some cells, few cells, which are around here, the central area, indicating that may be neurons. Actually, their position is not very clear here. That can be better seen here at later stages. They are at the inner nuclear layer, just on top of the inner plexiform layer. seemingly like they are amacrine cells. And what Camila did was to do a morpholino ablation of amacrine cells, specifically by injecting this combination of morpholinos to PTF1A, which is a transcription factor needed for amacrine cell differentiation. And what we see here, the cells disappear. These are amacrine cells here in green; they disappear completely. And we see here also a disappearance of expression of SLE2 in the retina. So what happens? This is also demonstrated here, where we see ablation of the amacrine cells with loss of SLE2 expression in the retina, but not in the brain. For example, in the floor plate. Actually, there is no clear phenotype in the retina. There are no layering problems. There are no very important problems, at least in axonal fasciculation or guidance inside the retina. And this also, there is a quantification here showing that there are no big differences in RGCs in the retinal ganglion cell generation. So that doesn't seem to be affected. But what we are trying to look at now, and this is something that I ask you to come and see eventually Camila's defense soon. Soon it will be on in October, by the end of October. She will show some results also on related on combination with other signals that could be involved in retinal ganglion cell differentiation and actually orientation of these cells. So it could be a role in retinal ganglion cell orientation, but that's something more subtle, and it's only seen when you combine that with other signals like blocking laminin, for example.
Okay. I have one more question. You don't find any evidence of the effect of the organization in the distal tracking mutants? It's not my topic, so I suppose there could be other guidance cues to organize the axon in that position, right? And which are that cues or different from SLE2?
Well, that's right. Actually, slits, they don't seem to be having much of a role there at that portion, and there is no expression. There are already known attractants probably making the axons to continue growing. One thing is that there is fasciculation caused by addition signals, so they will try to continue growing together and segregated. That's something that naturally they tend to do. And eventually some signals coming from the tectum could be acting. And eventually when they innervate retinotopically, they depend mostly on the F signaling pathway. So there is a differential expression of ephrins and F receptors in the retina, and then ephrins in the tectum, so attracting different axons to different portions. So that what makes the axons to go exactly to where they have to contact the target neurons in order to maintain this retinotopic order, which is very important for the visual field, for generating a good visual field.
Okay, well, thank you, Flavio. Now,
thank you.
It's a pleasure to introduce the third speaker, Dr. Kerstin Schmidt. She studied medicine at Frankfurt University, Germany. She obtained her doctorate in neurophysiology, Max Planck Institute for Brain Research, and her physician license in ophthalmology. Her current positions are full professor of systems and neurophysiology at the University of Rio Grande do Norte, Natal, Brazil, director of the Bryan Institute, University of Rio Grande do Norte. He has published more than 30 papers in journals of high impact in the field. Her research is focused on sensory neurophysiology of vision, in particular, cortical representations, intrinsic and feedback connections in the visual system, postnatal development of the visual system, and cortical microcircuitry. The title of her talk is Studying the Evolution of Visual Cortical Circuits in Outstanding Species. Kerstin?
Thank you very much. Good morning, everybody. My name is Kerstin Schmidt. I want to thank the organizers of the FALAN 2021 symposiums for inviting me to this very interesting symposium. and I'm the head of the Neurobiology of Vision Lab in the Brain Institute at the Federal University of Rio Grande do Norte in Natal, and I'm currently also the director of that institute. And in my lab, we are studying this outstanding species here, the agochi, and I will explain you in a second in which context this species is outstanding and why it's interesting to study the representation of the visual environment in its cortex. So let's get started to put us on the same page again. Which features actually matter in the visual environment for us to represent in our brains? And of course, you might say it's color. But for many animals, color might not be so important. What is important for all of us are contours, contrast contours, and they have certain orientations, and not only in artificial environments as seen in the picture, but also in kind of natural environments as behind in my background. So let's try to remember, what do neurons in the primary visual cortex actually respond to? They respond to contours of a certain orientation, and they don't do so in many mammals which are very similar to us and have frontal eyes like monkeys or cats, like scattered around in the visual cortex randomly responding to this orientation or that one. No, they cluster together in big groups of similar feature preferences, making up these domains which you see here. So if we stimulate an animal with contours of a certain orientation, many of such domains, which all consist of thousands of neurons, will be activated at the same time. Those domains actually extend through all cortical layers, raising up the justification to give this the term color. If we now join all these orientation columns together by vector summation, and if we have studied them in discrete steps, we end up with these colorful maps, which you all have seen already in neuroscience books, around with these points here, which look like pinwheels, and around these pinwheel centers, the orientation preference of the neurons changes gradually. So these pinwheel-like maps have been found in many animals, as I said, in carnivores. Here an example, the ferret, in primates, also in nocturnal monkeys like the galago, and also in scandensia, which are remotely related to primates, but are no primates, and even have, like the tree shrew in this example, quite lateral eyes. So the question arose, is it a universality of the cortical columnar design? This is what orientation selective neurons do. Do we need these columns to see, or is it a structure without function which emerges because of the way we study vision or the visual representations? And of course, there are numerous theoretical models why orientation columns emerge out of orientation selective neurons in two-dimensional structures. And these models, and this is only one of them, assume always that there are selective long-range connections connecting neurons of similar orientations. And at the same time, we know there are also short-range connections which are less selective, and a lot of inhibition which has a shorter range, usually than the long-range excitation. If you put all this connectivity in equations, you come up eventually with this pinwheel-like layout for orientation selectivity. If you fiddle around with the weighting of the connections, you'll eventually come up with different layouts, but still orientation selective neurons. Kulakov and Shklovsky said that the singularities in the pinwheel maps, they serve to optimize actually the wire length. So it's a way to economize length of the wires. So as time went by, also the rodent community entered visual neuroscience, because rodents are a very important model for neuroscience, as you know, for a lot of neurological diseases. With the emergence of newer techniques like the two-photon imaging and the calcium imaging, it was possible to visualize orientation maps in very small scale. And what you see here is that actually the cats show the pinwheel-like patterns. Also, if you zoom into a pinwheel, and they have very orientation selective neurons in the center of the pinwheels. But if you look at the rodent visual cortex, it's not that the neurons are not orientation selective, but they don't form these pinwheel-like maps. They rather form something which got the name salt and pepper design. So to date, there are two distinct layouts of orientation selectivity we know of, the salt and pepper layout and the pinwheel-like layout. So it's an interesting question, actually, what in evolution made these two different layouts appear, or are they only two layouts? So which factors could determine the layout of orientation preference map, apart from the ones the model physicists put into their equations? So, of course, the habitat of the animal might be important. Tree shrew, yeah? But a tree shrew can also live in the grassland, and all of them have pinwheels. It could be important that the animal is diurnal or nocturnal. It could be important that the animal is a predator, like most of the animals with pinwheels are predators. But that's why it is important to study the extreme species. The grasshopper mouse is a rodent, but at the same time it's a predator. No, it doesn't express pinwheel-like layout. Lateral versus frontal eyes could be important, binocular overlap. But there are also extreme species which have less binocular overlap and still have pinwheels. The retinal layout might be important because you might remember that all the rodents, or also the order of the clearers, the rabbits as well, have their eyes very lateral, and they have a horizontal streak instead of a fovea, so that might be important. Visual acuity might be a factor, so it might be important to optimize wire length and optimize maps and produce columns for visual acuity. And finally, the cortex size, something which also already came out of the early models, which modeled the emergence of the maps, who said, yeah, if it comes to a certain cortex size, it might be important to express this highly ordered design in order to economize wire length. So which gaps in the mammalian tree we still have to fill in order to answer those questions? Which outstanding species which are at the corners of the tree we have not yet studied? And the agouti came into our mind because it's the rodent with the so far largest investigated visual cortex in terms of orientation selectivity. And when we started this, its cortex size, its cortex is almost at the size of the visual cortex of a cat. And it's a rodent, and it sees actually very well, and is a diurnal animal, as opposed to other rodents, which are nocturnal animals, most of the rodents we knew of at that time. This is just an excerpt of this little movie. Most of you South Americans know what an agouti looks like and what it likes. It likes to eat fruits and seeds and nuts. And it's a ground-dwelling animal, not so much like a squirrel, but it can jump very high. And it uses both hands to feed. So at the time we came across the agouti, we knew the following. So it has a horizontal streak and not a fovea. Its B1 extends about here at the occipital pole of the brain, and the size is comparable to the V1 size of a cat. Its receptor fields are quite small, and which renders it a highly visual animal, and they are quite small throughout the horizontal streak representation. When we studied the orientation selectivity of the neurons, we saw that they have highly orientation selective neurons, comparable to other rodents, not as high as carnivores and primates though. However, when we started to do imaging on these animals and try to visualize the orientation preference map produced by the stimulation of contours of a certain orientation, we observed that cortex responds very well because neurons are orientation selective. However, those neurons do not seem to be clustered in groups of neurons and do not seem to be produce orientation preference columns. Rather, there's a homogeneous activation all over the place. And as you might see, there are some orientations which the Aguchi neurons seem to like more than other orientations. In fact, these are the more horizontal, oblique orientations. And this is something which was known from other animals with a horizontal streak, also like rabbits, that they have actually a bias for orientation preference around the horizontal, which might be related to the horizontal streak organization of their retinas, as opposed to a cardinal bias, which is often found in animals with frontal eyes, or in animals with an pinwheel-like layout, like the cats, the monkeys, or the ferrets. A preference for both vertical and horizontal orientations. On top of this, we found that, as actually was reported already for mice and rats, the Aguchi organization of orientation selective neurons is not so random as it seems at first. So if you record in depth from what is supposed to be a minicolumn, a structure which extends through all 6 layers, you see that the orientation preference of the neurons doesn't change so much. It also doesn't change around the same multi-unit. So the likelihood that you encounter a neuron of a similar preference close to another one is actually quite high, also in the Aguchi. This was also observed in rats and mice. So there is a clustering of neurons of a similar tuning in a distance which is smaller than 50 micrometer. But as you go far away from that, then the similarity goes close to random, justifying the term salt and pepper. However, so we can say now, yeah, we studied the big cortex, and the big cortex didn't impose the need on orientation selective neurons to express pinwheel-like maps. What mattered in this case was probably that it was still a rodent. So how about if the cortex gets infinitely small? And this is a study which appeared at the same time, interestingly, studied the cortex of a very, very small primate, the mouse lemur. And the mouse lemur is a nocturnal animal, and it lives also in Madagascar, like the galago. And it's actually not so, its visual acuity is not so sharp like of a big monkey and other primates. And interestingly, it produces really nice orientation preference map. And then an interesting question in this discussion was also, okay, so maybe the rodent columns, they are extremely small, they are just compressed, but they are nevertheless columns. And as you can see here, even though this cortex is quite small and the animal is extremely small, orders of magnitude smaller than the big monkeys, like this size, its orientation preference columns cannot be infinitely compressed. Rather, they still obey certain rules, like as, for example, the density of the singularities per hypercolumn seems to stay the same, independently how big the cortex is. So a large rodent and a tiny monkey confirm actually that the expression of orientation preference maps is invariant of the size of the cortex. It has to do with other rules. So let's re-evaluate by adding these 2 new species into the picture, the predictive value of some biological signatures. So the ones we can exclude now is actually something which was studied before, the relationship between the size of the visual cortex and the retina. As you can see here, actually, this relationship doesn't predict so well which layout is actually adopted, because we have species here which have a large visual cortex, like the Aguchi, and also a large retina. On the other hand, binocular overlap. So a question I'm always asked is, yeah, but all the rodents have their eyes on the side. If you look at the binocular overlap question, you see also that the tree shrew has actually quite low binocular overlap, as have some rodents, and it expresses orientation preference map. However, which seems to be in the race still as a factor predicting the layout, is the density of the neurons in V1. As it happens to be, and this was also already known, is that when rodents increase their cortex size, they do actually increase the number of neurons at a different power function as do the monkeys. This makes that they have actually quite low cell densities in the visual cortex, as opposed to a monkey or carnivore with the same size cortex. Another factor which is also still predictive is the central to peripheral retinal ganglion cell ratio. So this is something which is related to the horizontal streak of the retina, but not entirely. And if you compute the central to peripheral retinal ganglion cell ratio in all of these animals, you see that all the animals which express no maps fall below this 9. But what seems to be really predictive is the order. So if you belong to a non-glerous species, your chance that you express pinwheel-like layout is quite high. So let's add the 2 new outstanding species into the picture. So we have the smallest monkey and we have the so far largest rodent investigated in this question so far. And they both express the layout of their order independent of the size of the cortex. So what can we conclude? So there are still 2 distinct layouts for orientation selectivity. So orientation columns do not seem to be able to be compressed. And at the same time, there's nothing else between. So you have either salt and pepper or you have columns. Although we still cannot completely exclude that there are other layouts, which we might discover with different imaging techniques. Predictive values are still the order, the central to peripheral retinal ganglion cell density, and possibly the cell density in B1. It is possible that the salt and pepper layout is the ancestor layout. So we could go into this question by studying other extreme species to elucidate that question. It is possible that orientation columns, or the colorful pinwheel-like maps as we know them, have developed independently in the different branches of the tree as a punctured equilibrium. And sometimes this might not even be optimal. It might have been more optimal for the little cebus monkey to adopt a salt and pepper layout instead of sticking to the columnar layout. And in this sense, last but not least, it might be actually interesting to revisit the question whether the column which started as a functional column is actually a structural thing without function. But we don't think this is the case. We think evolution didn't spend all this energy to develop that layout if it doesn't serve a function. Last but not least, I want to thank you for your attention, and I want to thank my group in Natal who helped to implement the Aguchi model together with me. First of all, Dado Ferredo and Sergio Conde. And I also want to thank the UFERSA, represented by Moacir Oliveira, who bred the animals and helped us getting hold of them, learn how to handle them, and also Fridtwolf in Göttingen, who initiated the project of the evolution of visual cortical circuits in our lab. Thank you.
Well, thank you, Kerstin. It was a wonderful talk. I have one question. Could the different layouts for orientation selectivity in rodents and primates functionally relevant for visual processing?
Yes, that's an interesting question. So, of course, this was troubling the scientists ever since they discovered these layouts. Also because the study of vision in rodents is relatively recent compared to the study of vision in the binocular, in the more frontal-eyed animals like us and the primates. Mm-hmm. And in the last years, there's a lot of evidence from with the newer techniques from the rodent community that they actually have similar kinds of connectivity implemented in their cortex as have the primates, like selectivity for the same orientation, anisotropies, which would technically enable them to do the same kind of gestalt processing like the primates do. On the other hand, there are newer behavioral data which actually show that primates might actually use other figure ground segmentation strategies, so that actually there is a reason to develop a different layout, which might at the end serve some purpose. So like I said, the evolution didn't invest all the energy to make this complicated layout if it wouldn't be more efficient for the things which are important for the primates.
Okay, thank you. I have one question. My question is not my topic, but you show that you can describe two types of layout organization, pinwheel and salt and pepper. And you say that there are the two ones, but if you use other image techniques, you can find something in the middle. I understand it's correct or not, or
I don't know. It's the thing is that all the high resolution imaging techniques, as you know, they study very, very tiny volumes of cortex. So we know a lot of the fine resolution in the mice and in the rats, but we don't know, for example, the fine resolution in the agoutis. On the other hand, if we take the lower resolution imaging techniques, the classical ones like I used, like the intrinsic imaging and the voltage sensitive dye imaging, you lose the fine resolution. But it could be that there is a larger scale regularity which has, because of the low resolution of the imaging techniques, yet escaped our scope. And the hope would be to have a larger window and then see the neurons at finer resolution and see the regularity which escaped the other techniques.
Okay, well, thank you. Thank you so much. Well, the last speaker, it's a pleasure for me to introduce Jimena Sierra Alta. Jimena is a biochemist. She obtained her PhD in science, major in biology, University of Chile. Her current positions are professor and principal investigator, University of Chile, chair of the Master in Neuroscience Committee, associate professor and principal investigator, Institute of Biomedical Science, School of Medicine. She has more than 45 publications in her career. Her research is focused on mechanisms of synaptic transmission, studying the energy source for the synaptic activity and the neuronal glia energetic relationship and genes involved in neuronal morphogenesis. The title of her talk is Drosophila melanogaster, Neuromuscular Junction as a glutamatergic synapse model. Jimena?
Hi, I'm Jimena Serralta from Universidad de Chile in Santiago, and I will present today the Drosophila neuromuscular junction as a model of a glutamatergic synapse. We know that the neurotransmitter release is tightly coupled to presynaptic calcium kinetics. Thus, an action potential triggers the entrance of calcium into the button, and that produce the release of the neurotransmitter through the fusion of the vesicles, of the synaptic vesicles. The fusions is produced in the active zones, which are specific places where the machinery for the release is located, together with calcium channels and other proteins that are necessary for the activity. So the release of the neurotransmitters activate receptors in the postsynaptic synaptic side that in turn will open channels that will induce a depolarization in the postsynaptic. So thus we can record currents from the postsynaptic, and we can record evoked currents induced by the action potential, asynchronous release, asynchronous currents that are the product of the residual calcium, and spontaneous currents, product of the calcium, the resting calcium. The regulation of calcium in the button includes the entrance of calcium through the voltage-activated calcium channels, as well as the extrusion of the calcium from the cytosol through plasma membrane carriers like NCX or the pump PMCA. Also through the calcium pump in the endoplasmic reticulum. Thus, the presynaptic calcium increase due to the action potential, and the extrusion and buffering of the calcium efficiency. It's the key regulator for the magnitude, synchronicity, and kinetics in general of the neurotransmission. In Drosophila, PMCA has been shown to be the most important mechanism to extrude the calcium, the bulk calcium. PMCA is present in the neuromuscular junction, and in the mammalian system, alteration of neuronal expression of PMCA is associated to pathologies, neuropathologies in humans. Also, in the C-terminus of the PMCA proteins, there is a PDC domain or PDC interacting domain that could interact with MAGOC proteins. So, for a long time, in my lab, we have been interested in the role of the MAGOCs in the synapse. We explore and demonstrate several features of the DLG MAGOC in the Drosophila melanoras in the postsynaptic side of the synapse of the neuromuscular junction. And lately, we have been more interested in the presynaptic side, and we already know that DLG interact with some components of the voltage-gated calcium channels and regulates its location in the active zone. But we also know that the residual calcium in certain conditions is altered in DLG mutants. So we wanted to know if PMCA was associated to this deregulation or dysregulation of the calcium, of the residual calcium. Scaffold proteins of the MAGOC DLG family are very important in synaptic function, and they are known proteins to produce clustering of receptors, mainly in mammalian. The most known protein is PSD95, which binds to NMDA receptors and produce the clustering of these receptors in glutamatergic synapse. In Drosophila, there is only one gene called DLG, and it's known for structure of the synapse and be important for its function. It has 3 PDC domains, and these 3 PDC domains are able to bind the carboxyl terminus of multiple proteins in the membrane. In mammals and in Drosophila as well, DLG proteins are present pre- and postsynaptically in all glutamatergic synapses. Drosophila is a well-known model system to study multiple research lines, especially in genetics and in neuroscience. We have used Drosophila for behavior and genetic and finding genes that regulate genes associated to disease, to human disease. But the neuromuscular junction have been used for a long time to study glutamatergic synapses, and they share a lot of features with mammalian glutamatergic synapse. They are structured in boutons, and these boutons contain multiple active zones that can be labeled very specifically with markers like BRP. So to study the role of presynaptic DLG in the calcium excretion, we use third instralave, and we downregulate the expression of DLG or PMCA presynaptically using the UIS GAL4 system and two different GAL4, one pan-neuronal and one motor neuron specific. And also we use the DLG mute. For recording the currents, we use two electrode voltage clamp in the muscle 6 and in normal LCHEL 3.1 extracellular solution. We started this study by looking at the result of the accumulation of calcium, the bulk calcium, after 150 stimuli at 20 Hertz. This produced an increase in calcium in the button that after the stimulus is stopped, maintains releases, asynchronous release until the calcium goes to resting levels. This is the result. So in wild type, you see that there is a small amount of this asynchronous release, but in the LGS97, this release is much more increased. This is the same phenotype that in the decrease of the downregulation of PMCA or DLG produced, only the downregulation is in the motor neuron. And with the two different GAL4 lines is the same phenotype. When we quantitate these results using the total amount of charge transfer, we can see that all the conditions are significantly increase the asynchronous release. So neuronal DLG decrease in the neuromuscular junction produce an increase in the synchronous release, which is a result of the increase in resident calcium surrounding the active zone. In order to determine if DLG is able to alter the resting calcium in the button, we measure the frequency of spontaneous release, which is a reflex of this resting calcium. And so in wild type, there is a frequency that is increased when it's downregulated DLG or PMCA at physiological calcium. However, then this is quantitated. However, when the calcium, the extracellular calcium is 0.2 millimolar, we only observe this increase in the downregulation of PMCA, suggesting that although DLG is important for the spontaneous release frequency modulation due to the resting calcium, it's not as important as PMCA. Thus, neuronal DLG decrease increases the asynchronous and spontaneous release, and which is a reflex of the presynaptic calcium homeostasis that is altered. And the resting and residual calcium alteration could be due to the PMCA misplaced or decrease in amount. Look for in confocal and STED microscopy, we look for the presence of PMCA and of PMCA in the presynaptic component. To do that, we had to downregulate also the PMCA and DLG in the postsynaptic side, because as the membranes there are much more abundant, the signal for PMCA and DLG are very strong. To see then the PMCA and DLG in the presynapse, we had to do that. So these are pictures of buttons where the PMCA and DLG were downregulated in the postsynaptic side of the, and you can see clearly now the presence of PMCA and DLG in the presynapse. Here is PMCA. When we do the colocalization of PMCA and DLG, we see that these are form clusters, both DLG and PMCA, that are highly colocalized. So to determine the amount of DLG, the amount of PMCA in DLG mutants, we made a ROI around the membrane, the presynaptic membrane, and quantified the amount of PMCA. And we didn't find a difference between controls and DLG mutants in the total amount of PMCA. So we looked for more subtle alterations, looking at how they distribute. And you can see clearly that the wild type show these clusters of PMCA that are not present in the in DLG mutants. So, or the clusters are much smaller. And when we quantitate that, or to quantitate that, we again did the ROI only in the membrane, and we saw that the distribution of the size of the clusters is altered. So in DLG mutants, most of the clusters are very small, while in the while in the wild type, there are several clusters that are big that are completely absent in the DLG mutant. This is significant, the difference in the distribution. So DLG mutant alter the presynaptic localization of PMCA in the membrane, where the clusters that are characteristic of PMCA, big clusters, are now disappear. So our model is that DLGA and PMCA form, or DLG helps to form cluster of PMCA that help to do a much more efficient work of PMCA. When calcium enters, then efficiently is removed by the PMCA and regulates the synchronous and spontaneous releasing. So this is all. Thank you very much. I want to acknowledge Andres Kohler, who was the one that did all the work, and of course all the people in my lab, and particularly our collaborators Ulrich Tomach and Oliver Kobler from Leibniz Institute at Magdeburg. Thank you.
I have one question from Osvaldo. Can you avoid the increase in asynchronous release due to lack of PMCA with AM buffers?
Thank you for the question. No, we haven't done that, but we have rescued the mutant phenotype with re-expression of DLG in the presynaptic side, and it completely rescued this the phenotype. Probably the phenotype will be rescued with these buffers. I mean, it should be. I will expect that, but we haven't done that, the experiment.
Okay. And I have one more question. What you expect to be the consequence of DLG mutation in the adult animal? Do you expect a consequence in brain that display a majority of glutamatergic synapses?
Well, the adult animal in Drosophila, just for the vertebrate people, the brain is not mainly glutamatergic, but cholinergic. So, but still there are several neurons, not the majority, but a lot of neurons that are glutamatergic. So, on the other hand, DLG is expressed in the whole brain. I mean, most of the neurons express DLG as invertebrates, although they are not glutamatergic. So I expect that we'll have a phenotype. So we have done that in DLG mutants, and DLG mutants age faster than the normal wild type flies. But we don't know if it's that specific for glutamatergic neurons or it's because it's in all neurons. On the other hand, I expect that as DLG produces this increase in resting calcium and also in the amount of calcium that is accumulated, I will expect that the neurons will have more oxidative damage and maybe probably a neurodegeneration that is faster than in wild type. So I will expect in adults and maybe invertebrates that the aging process is faster and maybe that the neurodegeneration could be faster too.
Okay, thank you. Thank you so much.
Thank you.
Okay, we are all together now. Thinking about the title of the symposium, only to open the discussion, I would like to ask to all of you about the animal models that you chose to do your research, the reasons and the advantages and disadvantages, because I think it is in the spirit of the symposium to make this point about that. So who wants to answer first? Juli?
Okay, yeah, well, I got thinking about function of the brain and the diversity of models, also following Kirsten's talk, because sometimes when you focus only on the typical lab models, you get like the general picture and the definition in the physiology book. So you know that vertebrates have the orientation columns, and unless you go out and find new animal species with different type of environments, I think the general knowledge is kind of well, it is limited because we don't know the diversity that can really exist. In my case, working with crabs, well, has the contra that there is not much knowledge about. So every time we want an answer to a question, we have to do the experiments or the techniques to do that. But I think that also gives you an opportunity to expand and do different things. But especially selecting models that you can connect with their behavior and the ecological needs of that species. It's very rich because it can tell us, in my case, considering the network behind visual processing and leading to behaviors in the natural world, it can give you clues of what parts of the systems are more adaptable or changeable than others. And I think that is also nicely presented in Kirsten's talk as well.
Mm-hmm. Well, thank you. who wants to answer?
Yeah,
Kerstin,
I can say something. So about the model. So I really appreciated hearing of all of your models, and it's actually something which I do not enough to explore the non-vertebrate communities, and I learned a lot, especially about our world. So it's like vertical and horizontal dimensions are important, and they are for all of them, but which is quite obvious. So to your question, Eugenia, so why choosing the model? Yeah, the advantage of our model was that it's really the rodent which has the largest neuronal quotient known so far. So it's if one of the rodents would have been able or would have had the space and the capacity to express this more modular layout, it would have been this animal. So that was the obvious advantage. We had a very visual animal, which was diurnal also, because there was also always the critiques on the rats and mice studying their vision, is that they are nocturnal animals. And the other thing is, yeah, it's the same size visual cortex as a cat almost, and so that was the big advantage. And I think having not seen these kind of columns there, pretty much responds to the question. It wasn't a problem of size. And the disadvantage, of course, is, or what is the advantage of the smaller models is that you can combine a lot of things in one thing. So you have the high resolution, you have circuits which are maybe not less complex, but you can get them from different perspectives, which is much more difficult when you have the huge animals. And if you put behavior into the experimental paradigm, it gets even more difficult, and because then there's attention and then there's some kind of cognition we have no idea of, and yeah, and so that's why I think it's extremely interesting hearing about this invertebrate models and the crab and the optic flow. I think this is a big advantage, that you can do the behavior and study the direction selectivity at the same time in a model which is maybe more predictable.
Well, thank you.
So, I can.
Yeah,
yeah.
Obviously.
I think that it depends a lot about the question. So I think that the question drives you to a specific model finally, no? As Kerstin with the agouti and Julieta for the crabs. I mean, it's very specific. In my case, it was kind of the other way around in the sense that I didn't want to work with rats and mice because it was strong for me, the need not to work with them. But I wanted to work with some animal that you could do things in vivo. So it's why I moved to Drosophila. So at the beginning, I started working with rats and mice and also with chicken. But finally, I got very much in comfort with the flies, because you can do in vivo not only recordings, but also behavior and genetics and microscopy and everything. And the genetics is amazing. So also because I'm in the biomedical field, and for biomedicine or for biomedical research, you need to stick to a model and then compare it to vertebrate, to mice, and then to humans. But you have to, I mean, you don't have to do it by yourself. You can do it with other labs that work with rat and mice, and then with humans. But for me to work with Drosophila has been also very important for the economic part, because it's so cheap to work with. And in Chile, we don't have as much money as the US and other parts of the world, but it's very Latin American need for cheap models that can be used and you can publish and you can find amazing things. So I love Drosophila.
Okay. Well, I think that is our case with the Neolese granulata with crabs. It's also cheap, and you can answer questions, and you can publish in good.
No, I think crabs are great models.
No, the thing is that you, if you have a good question and you can know to use that model, you can be
exactly.
I feel like. Okay, Flavio.
Thank you. Okay, well, my case is similar to Jimena's. My formation mainly comes from the cell biology side, and so I wanted to know about the cell biological, molecular biological processes in vivo, but mostly invertebrates. That was most interested in. The zebrafish is perfect for that because also has genetics, not as good as in the Drosophila, of course. It also has the optical accessibility, which is essential, and they are. I mean, if you look at them, they look more like, especially the embryos, they look more like invertebrates because of their size and the transparency, and so it's you can do a lot of things in vivo. Well, that is the main reason for working on that. Still, you cannot, of course, in any model, you cannot answer all questions. We also work with chick to try to compensate for some things. The chick is much more limited, but still gives us other answers. For example, it is an amniote compared to an amniote that is a fish, and that brings big differences. But of course, then you have the limitation again, limitation of like when I was saying of the economical part, which also is a good thing in the zebrafish compared to other vertebrates. Is rather low cost, let's say, and also you cannot, of course. It would be interesting to work in many other species, but then there is a big economical restriction because you cannot diversify too much unless you are extremely rich. So keeping on those two species and mainly on zebrafish has allowed us to answer many questions that were interesting for us.
Well, but I think because all of you mentioned, and I also think that we found the way to do good science using in our Latin American context, like with our restrictions in economics, and we found, and you are outstanding researchers, so it's possible to do that. Being here is like, I think that it could be a good message. Juli, I watched that you leave a question on the chat. You want to share, or?
Yeah, sure. I was thinking, well, some of the talks in this symposium a little bit made me think about the crosstalk between the morphology of the brain or the organization of the network and the actual function. So I would like to ask Flavio if they have tried measuring the visual capacities of the animals they have altered the chiasma, and to see if some function still resist the manipulation of the chiasma, and also because there is a lot of discussion about what is the actual significance of the chiasma. If it's only something that is morphologically useful to accommodate the axons and make them all the same size, or if there's something else that is basically needed for the physiology.
Thank you, Julieta. Yes, it's a very interesting question. Actually, we want to know the answer to that as well. We haven't done those experiments. We are actually with one person that is making some questions in the other chat now, Francesco, who's just 3 floors below us. So he's trying to set up a system for analyzing optomotor response in the mouse, and we are hoping to be able to adapt that for the fish as well. it would be very interesting. Fish are very normal in general. So the SLIT2 mutants, which are stable mutants, and we can keep them even as maternal zygotic mutants. So they have not even maternal expression of the gene. They are completely normal. They breed. They have all the behavioral features of a normal fish when you just have them in the tank. Of course, there's something else that could be happening. It could be we could have some visual acuity problems or something like that, which it would be nice to analyze, or of course directional problems or things that we cannot see just by feeding them or seeing them behaving in the tank. So yeah, that's a great question. And I was regarding the chiasm, I was seeing your double chiasm in the crab, and that's also very interesting, because then it will correct, it will like again make the things in the same direction. And it's a big question in evolution, of course. So why there is a chiasm? What is the advantage, if there is any, or if it's just an evolutionary, well, something that was left because the animals turned around from invertebrates to vertebrates, and then eventually we got that solution to still innervate properly. That's a big question, very interesting one.
Okay, Kerstin, I have a question from Francesco. Can you study classical dominant plasticity in your species? Already tried?
Yes, thank you. I briefly answered it there. Yes, in principle, you could study, but then you would enter a classical field again with a new species. I don't know if this is justifiable, because you have to work with developmental animals in development. And I think the big animals, monkeys, cats, so their time is over. But of course, it would be interesting to do so because it's a big rodent. And in principle, it's possible. It has a relatively large binocular field, although I haven't been there. It's supposed to be there. And so in principle, it's possible, but you would have to go into different things, breeding of the animals, and as Flavio said, so it's a question of feasibility as well, no? Because if you work with bigger animals, you have other timelines as well. The breeding is not so fast.
Okay. And I'm thinking because Julieta and Flavio talk about the functionality, and in your slide, the last point that you put there is that revisit the question of whether the functional column is a structure without function, or I find this idea very interesting. Could you explain, because I find there a relation, or I suppose that there is a relation about the structures and the function, and you say that it's possible, but you can explain it more in depth, or it's not related to.
Yeah, the thing is that, for example, as somebody raised the issue of the ocular dominance columns, they are more structural because, and this has all to do with the chiasm and with the crossing of the fibers and with the development of the visual system, and eventually they end up to be columns, and they have a structural basis because the fibers grew in differently. Like there's the classical experiment of the frog of Martha Constantine Peyton, which developed columns in the tectum eventually when they implanted the third eye. But on the other hand, we were talking here about functional columns, which appear by the way we study them. So there is not quite this strict morphology about them, even in the animals who have the pinwheel, like mosaics. It's not that you see, here's the end of the column, here starts the next one. There are the regularities, though, which may lie in the type of the long-range connectivity, which makes the patches, which you might not have in the rodents, which have the salt and papillae layout. And that's why I think there might be some structure in the function in the end, and that there might be actually a reason why this has developed. So apart from the microcolumns, which all of the animals have, which are more or less the same size, and are limit function to all of the systems which can be put on top of it. So I think, yes, it has to be revisited. And there were reviews and stories about this, where and they were always like fixed on the ocular dominance column. So there are animals who have stereo vision which don't have ocular dominance columns, also monkeys. So that's why we don't need them. But here we are talking about another columnar system, and maybe the question has to be separated.
Okay. Okay. Well, we are on time, or yes? No? Okay. Well, perfect. Thank you for all of you. I really enjoy the symposium. I learned a lot. It was amazing. So see you in the later activities of the meeting. And thank you for being here.
Thank you very much, Evelina.
Thank you all.
Thank you very much.
Bye.